The main theme of this presentation is individual differences in psychological processes and behavior. So I'm very interested in what makes people different from one another. And particularly, I want to talk about differences in what we call sociosexuality, which is the notion that some of us prefer to be in stable, long-term, monogamous, committed, romantic or sexual relationships, whereas others prefer to be in more promiscuous, less committed, romantic and sexual relationships. So I'm interested in the correlates of some of these differences in preferences, particularly in terms of differences in personality traits of people who have these preferences, differences in propensity towards risk-taking or novelty-seeking or sensation-seeking. I'm interested in correlates in terms of how competitive and cooperative people are, and so on and so forth. I'm also potentially interested in where these individual differences come from. Potentially they come from genetic influences, what we call embodied capital, and early life experience. Embodied capital refers to the notion that we're not all born the same. Some individuals are born small, others large. Some will grow up to be tall, others short. Some are born very healthy, others less healthy. Some will become very good looking, others less good looking. Some also are lucky to be born rich in a rich family, others in a poor family, and so on and so forth. We call all these things embodied capital because many of them are actually physical property of the individuals. For example, physical health, or even things like intelligence. Early life experience is important, so the environment in which we were raised affects the development of these preferences. I'm also interested in the potential physiological mechanisms that are underlying these preferences. The idea is that these preferences for relationships do not appear out of the blue, but they're linked to physiological mechanisms. So there's a strong connection between mind and body. So I'm especially interested in the potential importance of sex hormones, such as testosterone, and stress hormones, such as cortisol. I'm also interested in where these individual differences in preferences come from. So to understand this, we can use a branch of evolutionary biology called life history theory, which I will briefly introduce to you. And in this presentation, I will use two particular points of entry into this topic of individual differences in preferences. One is individual differences in circadian preferences, also known as chronotype. The fact that some of us like to get up very early in the morning and go to sleep early, others like to sleep in and then stay up until late. And I also would like to talk about individual differences in relationship status, which means individuals who are single versus individuals who are in relationships, long-term relationships, short-term relationships, or even marriage. So, if you're not convinced that these individual differences in social sexuality really exist, I will give you two examples that I think suggest that, for example, with regard to men, there are two types of men which differ from one another in physical appearance, so the faces, the way they look, in their bodies, their thoughts, what goes on in their minds, their behavior, and their preferences. So the first example is a study that my students and I conducted a few years ago. The subjects were a number of college students, in this case, male college students. We gave them different questionnaires, and one of them had a lot of questions about their interest in babies, interest in young children. So, for example, some of the questions asked, how are you interested in general in young babies? Do you find them cute? If you meet a woman with a young child at a party, would you approach and try to interact with the child? Also things like, are you thinking of having children later in your life? These are college students, so they're not married, they don't have children yet. And of course, some men say yes, others no. So there's variation in reported interest in infants. Then we had these young men watch a video with erotic content. It was nothing outrageous. It was a good-looking woman essentially taking off her clothes. We took a saliva sample from the subjects before and after this video, and we measured their testosterone in their. Typically, testosterone goes up a little bit after exposure to visual erotic stimuli. It turned out that the way they answered that questionnaire about interest in infants was associated with how much their testosterone increased. So those who said we're not interested that much in children had a spike in testosterone. They were very reactive to sex scenes. Those who said, oh, we're very interested in babies, not so much. Their testosterone does not increase. So this is very interesting because, again, these are young men. They're not married. They don't have children. It seems that there's already differences in attitudes toward children, which have a counterpart already in their body. So it seems that those who are not interested in children are more attuned to hormonal reactivity to obvious sexual stimuli. For example, a stranger, video of a stranger taking off her clothes, whereas others are not as comfortable or not as easily arousable by this. So I have reason to think that these differences emerge even before college age. We can't really measure testosterone in children, but I think that these differences in preferences that are revealed, for example, in interest in young children can be measured even in teenagers or younger children. The second example was another study that we did earlier. In this study, again, we asked these male college students the same questions about their interest in infants. Then we measured their testosterone, this time just baseline condition, one sample or two samples on average of their testosterone concentration in saliva. And then we took a photo of their face. Then we showed, okay, you can't see. Okay. We took photos of their faces. Then we showed the photos of these young men's faces to female college students that lived in a different city who did not know who these people were. And we asked these female students to rate these faces in terms of how attractive they were. We asked them to try and guess how much testosterone these men had in their body, whether a lot or a little, and asked them to guess how much they were interested in infants or young babies, a lot or not so much. And finally, we asked, so if you had to marry one of these men, can you rank them? These are the faces that you would be most likely to marry versus men with whom you would like to have just a short fling, just you meet them from one night, something happens, that's it. It turns out that these women were able to guess from the faces of these men how much testosterone they had and how interested in babies they were. So for long-term relationship, for marriage, they picked the faces of men who were highly interested in babies and had relatively low testosterone, whereas for the short-term thing, they picked the faces of the men who had a lot of testosterone and not so interested in infants. How did they do that? Well, I chose two sample photographs. These are not college students. And you can tell, for example, that the face of these two men is very different. One has a very large square jaw, the one on the left. This is a trait that is associated with testosterone, high testosterone. Having a large square jaw for a man means this man either had very early in life or concurrently has a lot of testosterone, whereas the man on the right has a more feminine type face shape. So you can guess from the shape of a man's face, for example, things like concentration of testosterone in their body. So these differences exist in interesting infants. They're reflected in testosterone profiles, and they're even apparent in physical appearance, such that other individual can guess. So I hope that this was more convincing than just making a statement. Now I would like to give you a brief introduction to life history theory, which is the evolutionary framework that we use trying to explain where these individual differences in social sexuality come from. Life history theory deals with the way organisms, so animals, but also humans, allocate their time and their energy to the various activities that comprise their life cycle, including their growth, their reproductive maturation, their reproduction, including both mating and parenting, and so on and so forth. The idea is that organisms don't have an infinite amount of resources. So if they allocate a lot of resources into one activity, they have less to allocate to another. So there are trade-offs. The best known trade-offs are those between survival, growth, and reproduction. So if organisms invest a lot in preserving their survival or in their growth, they have less to invest in reproduction, and vice versa. If they invest a lot in mating, they have less to invest in parenting. If they invest a lot in their current offspring, they have less to invest in future offspring. And if they produce large amounts of offspring, typically these offspring are poor quality, whereas if they produce one child at a time, they can produce a child that is very, very high quality. So these are the trade-offs. There are differences in different species in the way these resources are allocated to these aspects of their life cycle. And these differences appear as clusters of traits that are interrelated. So they include morphology, so body size, for example, physiological characteristics, reproductive characteristics, and behavioral and psychological traits. Variation in these life history strategies occurs along a continuum from what we call fast strategies and slow strategies. So there are some animal species that tend to be fast life history strategies, also known as R-selected. These are animal species that typically have small body size. They mature reproductively very early in their life. They have high fertility. They're able to reproduce very quickly, produce large amounts of offspring. They don't invest much in terms of parenting. They put out these offspring, tell them good luck. They have high mortality, both the adults and the offspring, and typically have short lifespans. Examples of this are insects. They do this, also small fish, and so on. At the opposite end, there are species instead that have very large body size. They mature very slowly. They reach adult body size and reproductive maturation very slowly. They don't have high fertility. They produce one offspring at a time. They invest a lot, and they live very long lives, like elephants. Human beings are more like elephants than like insects. Differences in life history strategies occur not just at the level of species, but also at the level of individuals of the same species. For example, human beings. And these different strategies appear in terms of, again, interrelated clusters of traits that include things such as self-regulation, tendency to take risks or to explore, aggressiveness, competitiveness, cooperation, mating, and parenting. The theory says that what gives rise to these individual differences in strategies is the environment, particularly the amount of resources that are available, the risk of mortality, whether the environment is predictable or not. In part, these differences are genetically based. In part, they come from the environment. In part, they're a result of an interaction between genes and environment. In human beings, fast and slow life history strategies have a number of psychological and behavioral correlates. So, for example, individuals who pursue fast life histories tend to have insecure attachment to their parents when they're little, but also to their romantic partners in adulthood. They tend to have a more individualistic, exploitative interactional style. So in relationships, they tend to be a little selfish. They're very ambitious in terms of status and dominance seeking. They score high in novelty seeking, risk taking, sensation seeking. They can be aggressive and sometimes violent. The males invest a lot in courtship displays. They can be sexually promiscuous. And in terms of personality traits, individuals like this tend to be more extroverted, a little neurotic, and tend to sometimes show traits that are known as the dark triad, which I will talk about later. At the opposite end, the individuals who pursue slow life history strategies tend to have secure attachments, they're more cooperative and altruistic, they tend to prefer monogamous, stable relationships, and in terms of personality traits, they tend to be more introverted, more agreeable. That figure is the cover of a book about life history that focuses on mechanisms. These are not just behavioral and psychological processes. They have also physiological, even anatomical counterparts. So important mechanisms for life history strategies are hormones, like sex hormones and stress hormones, because hormones filter the effect of the environment on the individual. Okay, so life history theory has been applied in many areas of psychology. For example, there are theoretical papers about risk-taking in humans, including in financial situations, which have used life history theory as a framework to explain differences in risk-taking. It's been used in studies of the family, for example, family dynamics. It's been used to explain differences in the timing of reproductive maturations in girls, to explain why some girls start menstruating much earlier than others. It's been used recently also to account for differences in psychopathology. There's a way to think about some forms of mental illness as being closer to fast life history strategies and others more to slow life history strategies. For example, autism as a general picture of slow life history characteristics, whereas things like psychopathy or the dark triad traits like Machiavellianism, narcissism, and psychopathy, they tend to be more fast life history. Even studies of substance abuse have been analyzed with a life history framework. Okay, so in this presentation, I want to address two general questions. One is whether individual differences in what we call chronotype, or morningness, eveningness, are a life history trait, which is in large part genetically determined or influenced. Particularly, I want to ask whether eveningness is associated with a fast life history strategy, while morningness is associated with a slow life history strategy. The second big question I want to ask is what we can learn about slow and fast life history strategies from studying same-aged individuals who are single versus individuals who are in relationships, including marriage. And I want to ask these two questions. Do these individuals who are single or in relationships have different profiles of sex hormones like testosterone or stress hormones like cortisol? And if yes, are these different hormonal profiles of these individuals the causes or the consequences of their different relationship status? So now I'm going to go through some studies that we've done, which address all these questions. So first, I'm going to talk about, in the first part of my presentation, about chronotype, or morningness, eveningness. This is the notion that some of us like to wake up very early in the morning, go to sleep early, and we have optimal cognitive performance early in the morning. I'm one of these people, one of these early birds. I get up at 4:30 or 5 in the morning. I read, I remember everything. My brain functions perfectly between 5 and 7. After 5 p.m., my brain is dead. If I try to read, I fall asleep immediately. At the opposite end, there are people instead that we call night owls, who like to wake up late, go to sleep late. Their optimal cognitive performance is not in the morning, it's late in the evening, even in the middle of the night. Of course, there are a lot of individuals whose preferences are intermediate, but these are the extremes. In fact, this trait is distributed normally in the population, such that most individuals tend to be around the average. So they're pretty flexible. They don't have a strong preference one way or the other. And the individuals who are at the tails of the distribution, which are fewer, show these extreme traits being either morning-oriented or evening-oriented. There have been a lot of studies about this trait, so we know a lot. We know that individual differences are stable across the lifespan. that they are heritable. So there is evidence of genetic similarity, for example, between parents and children or brothers and sisters. If you look at the graph on the right-hand side that has age in the bottom and a measure of chronotype, it's a numerical score that is obtained with a questionnaire that assigns essentially categories to individuals. You'll see that children tend to be early morning. So kids, little children, wake up early in the morning. But the tendency to wake up later and to stay up later in the evening increases at peaks after adolescence. So college student age, around 20, 25 years of age, we see a lot of individuals who have the late evening preference. This is the time where we also see the emergence of a pretty strong sex difference. So the night owl pattern is more common in males than in females. Then the tendency to be evening oriented tends to decrease with age. And also the magnitude of the sex difference tends to decrease up to the point where at the age of around 50, 55, when women reach menopause, the morningness pattern becomes more common and the sex difference disappears. So after 60 years of age, men and women don't differ anymore. We know, thanks to studies, some of which have been conducted here in the psychology department of the University of Bologna by people, some of whom are in this room right now, I won't name names, that there are differences in personality traits between extremely morning-oriented people and the night owls. So morning types tend to be more conscientious, evening types tend to be more neurotic, so a little more emotionally unstable. In some studies, even more extroverted. So our general hypothesis is that variation in chronotype is associated with variation in life history strategies, and particularly the fact that the hypothesis that the night owl patterns might reflect a fast life history strategy. So we expect these individuals to be more competitive, have greater propensities towards risk-taking, novelty-seeking, sensation-seeking, more sexual promiscuity, and so on. Whereas the early birds should reflect more of a slow life history strategy. We expect these individuals to be more cooperative, more egalitarian, more conservative, more sexually monogamous. So to test these hypotheses, the most obvious thing to do is simply to use questionnaires to see whether there is a relationship between a questionnaire that assesses these circadian preferences and a questionnaire that has been developed to assess the basic life history strategy of an individual. This questionnaire is called the Mini-K questionnaire. It was developed by a professor at the University of Arizona named Aurelio Figueiredo. The way people answer this questionnaire typically gives a portrait of a slow life history strategy. So the questions that are asked typically tap into aspects of slow life history strategies. For example, things like, while growing up, I had a close and warm relationship with my biological father. That's more of a slow life history aspect. Or I have to be closely attached to someone before I'm comfortable having sex with them. So I don't like having sex with people I just met, for example. So we found that, consistent with the hypothesis, that there is a close correlation between being early morning oriented and answering these questions in a way that reflect a slow life history strategy. Okay, so this is the first piece of evidence that just with questionnaires that being an early morning person or late evening is correlated with aspects of behavior, attitudes, preferences that cluster into different life history strategies. Then there's predictions about personalities. A lot of evidence was already available, that there have been studies of personalities in relation to chronotype available for many years. So previous evidence already showed that night owls, as I said earlier, score higher on extroversion, neuroticism, and these dark triad traits, which are narcissism, Machiavellianism, and psychopathy. Whereas early birds score higher on conscientiousness and emotional stability. We replicated some of these findings, so the graphs below show our own data. We found that night owls are more neurotic and early birds are more conscientious. So this also matches the idea of the two different life history strategies. Then we did several studies to look at the possible association between chronotype, risk-taking, and testosterone. The prediction is that night owls should report higher risk-taking and should have higher testosterone than early birds, because in one of these studies, we actually demonstrated that there's a correlation between high testosterone and high propensity for risk-taking. We did two different studies, one that was just behavioral, the other one that also had hormones. The first study was 172 adults, males and females. We used a questionnaire to measure risk-taking called the DOSPERT risk-taking scale. In the second study, it was 170 business school students. These are master's students in business administration, males and females. Here we just asked a simple question about risk-taking, and we measured testosterone. So we published the results of these studies in 3 different papers, some of which are just behavioral, others also have hormones. This is the DOSPERT risk-taking scale. It has 30 different questions subdivided into 5 different domains of risk-taking. There's ethical risk-taking, so questions such as, would you pass off someone else's work as your own? Would you be happy to plagiarize work? Or would you have an affair with a married man or woman? That's ethical risk-taking. Or financial risk-taking. Or health and safety risk-taking, like riding a motorcycle without a helmet, or sunbathing without sunscreen. Recreational risk-taking, like bungee jumping off a tall bridge. And finally, social risk-taking, which you can ask questions about, such as disagreeing with an authority figure on a major issue or speaking your mind about things. This I have some doubts about. This is not just a risk-taking. It's also taps into things like respect for authority, impulsivity, and things of that sort. Anyway, these are the data showing differences in chronotypes. So individuals are divided into 3 groups: the early morning, the night owls, and those that are in the middle, in relation to the 5 domains of risk-taking. As you can see, for 4 out of the 5 domains, the night owls score higher than the early birds. The ones in the middle are in the middle. The social risk-taking has an opposite trend, where the early morning people score higher. Again, I don't think it really measures risk-taking. So by and large, this is consistent with our prediction. We also replicated a well-known finding that men in general score higher on risk-taking than females. This could be in part biological, because men have higher testosterone, but in part is also cultural, because some societies and culture encourage men to take risks and instead discourage women from taking risks. So it's not just biological. Anyway, when we divide the risk-taking and the chronotype classification in relation to gender, we get this picture, which shows that in males, there's no difference in risk-taking in relation to chronotype. So the early morning and the late evening men are similar, but there's a difference in females, such that the night owl females score higher in risk-taking than the early morning females. In fact, the night owl females are no different than the males. So the basic result is that the early morning females are lower than everybody else, and everybody else is more or less similar. In terms of testosterone, there is not an effect. We see that the 2 groups of men have much higher testosterone than the 2 groups of women, but there's no difference in relation to chronotype. There is, however, another study conducted by some researchers in Germany, which did find a difference in testosterone in the direction that we expected, such that night owls have higher testosterone than early birds. This was done just in men. So it's not clear whether really testosterone differs in relation to chronotype, because there are 2 studies with conflicting results, so we need to do more research. Next, I'm going to show you the results of another series of studies in which we looked at the possible association of chronotype with other psychological and behavioral characteristics. There's behavioral and in some cases physiological data. The first variable of interest is what we call the present orientation versus future orientation. Whether you're a person who likes immediate gratification versus somebody who can delay gratification. You might have heard of this famous experiment called the marshmallow test, done by a psychologist named Walter Mischel, who died a few months ago in New York. He presented young children with this little marshmallow, which is something that they really like. He put it in front of them on a desk and he said, You can have this now, but if you wait a few minutes, hours until tomorrow, you'll get more, or you get something else. And some kids just cannot wait. They had to grab it and eat it immediately. And others instead were happy to wait. And then he followed these children for years when they became teenagers, adults, and so on. And he claims that how they responded on this test predicted all kinds of things, predicted their outcomes in work, in relationships. So the ones that can delay gratification in general are more conscientious, they meet their deadlines, better organized, and so on and so forth. So there is a questionnaire that can be used to assess this. So we didn't do the marshmallow test. We used this questionnaire called the Zimbardo Time Perspective Inventory. It has 56 questions that ask things like, I do things impulsively, or I'm able to resist temptation when I know that there's work to be done. We actually did 2 separate studies using this questionnaire, but we found the same results. We found that early birds are more future-oriented. They can delay gratification. The night owls are more present-oriented. They're more impulsive. They just want to grab the reward. That graph shows it's just a yes and no measure of whether they're future-oriented, and it shows that the early birds are much more future-oriented than the night owls that have very low scores, and the others are in the middle. Then we did another study with a simple questionnaire about how competitive men are in relation to other men in a situation that is related to courtship or possible beginning of a romantic relationship. Just to make a long story short, we found what we expected, which is night owl men are more sexually competitive with other men than the early bird men. Then we did something a little different. We had reason to expect the night owls to be competitive. And so we hypothesized that they would be more interested into social status, and that they would also perceive themselves to be of higher status than the others. So we used this test called the MacArthur Scale of Self-Assessed Social Status, which involves showing the subjects that little drawing of a ladder. And we tell the subjects, imagine that this ladder represents the hierarchy in the group of friends to which you belong. So these are all your friends, your colleagues, you could be the students in the classroom. Place an X on the step of the ladder where you think you are. So are you the most popular student? Are you at the bottom? Are you in the middle? Tell us where you think you are on the ladder. We found that early birds report being of lower social status than the night owls, which is consistent with what we expected. Then we measured cooperative tendencies. We used 2 different tests, one called the prisoner's dilemma. It's an economic game in which people have to cooperate with each other, 2 players. One is an ultimatum game in which one individual offers an amount of money and the other one has the choice to accept it or reject it. In the prisoner's dilemma, morning types were more cooperative, the men especially, not the women. And in the ultimatum game, they were more likely to accept low monetary offers. So this suggests both results that early birds are more cooperative or more socially tolerant, whereas the others are more competitive. Finally, we get to the bottom of our hypothesis, which is whether there are differences in sexuality between these 2 groups. In our study, we didn't have a lot of data on this. So what we did, we asked young men to tell us how many different sex partners they had in their life. So these are men around 25 years of age. So we had 68 night owls, 21 early morning males. There was a difference, but it's not strong. So on average, night owls told us that they had, on average, 11 partners. There was an exception. There was a guy who told us he had more than 1,000 partners. We did not believe him. We excluded that data point. But that says something about the personality traits of the night owls who like to be grandiose. The early morning males, on average, had 6 partners. There's another study done by, again, the German researchers with a much larger sample size, which finds stronger evidence that night owl men have higher number of sexual partners than early birds. We asked our subjects to tell us whether they were single or in a relationship at the time we did this study, and discovered, this is on the y-axis, the percentage of people who told us they were single, males and females. And as you can see, the night owls, both men and females, report a higher probability of being single. So there were more singles among the night owls than among the early birds. And this is something that I will explain and talk about later in the second half of my presentation, which I'm almost there. We did another study using a questionnaire that taps into social sexuality attitudes. It's called the multidimensional social sexual inventory. Essentially, this questionnaire will look at whether people prefer to be in long-term, stable, monogamous relationship. This is called a long-term mating orientation versus short-term, more casual, more promiscuous relationship. This is called a short-term orientation. So we found that, consistent with the hypothesis, the night owls are more short-term mating oriented than the early birds. So they're more likely to say, oh, I prefer short-term, more casual, more promiscuous relationships. And because we had data on these same subjects, on their time orientation, whether they were present-oriented or future-oriented, we were able to show that the association between eveningness, being a night owl, and being short-term mating-oriented is mediated by immediate gratification. So they prefer those kinds of relationships because they don't like to wait. So if they meet somebody they like, they're not willing to wait a long time until the relationship is settled to progress to the stages of the relationship. They want their gratification right away. So let me summarize the results that I presented so far. There's questionnaire-based evidence that night owl and early bird phenotype are associated with fast and slow life history strategies, respectively. In females especially, night owls score higher in risk-taking than early birds, and as high as the males. Differences in testosterone need to be further investigated. It's not clear. We found that, consistent with life history framework, night owls are more present-oriented, more extroverted, impulsive, higher in risk-taking. The early birds are more future-oriented, more introverted, conscientious. They can delay gratification. Present orientation mediates the association between, present orientation mediates the association between eveningness and short-term mating. We need more data on sexual behavior. What we have is insufficient, but the preliminary data that are available only for men indicate that night owl men tend to prefer these short-term, more promiscuous relationships. The conclusion is that being an early morning person or a night owl is associated probabilistically, so in terms of probability, with a number of physical, physiological, and behavioral differences that involve personality, attitudes, preferences, things like risk and novelty seeking, cooperation and competition, emotion and cognition. We also have data on differences in intelligence, which I didn't present, and social sexual behavior. So comparing these individuals that have different chronotypes can help us understand biological influences on mind and behavior and how these differences reflect differences in life history strategies. This is the end of my first half of the presentation. Now I'm going to switch gears and I'm going to talk about the relationship between life history, relationship status, and hormones. Our general hypothesis here is that being single in a short-term or a long-term relationship reflects both the choices that people make as well as the constraints that they have. But this is associated, we think, with different patterns of social sexual behavior, for example, being more monogamous or more promiscuous, different patterns of stress, and possibly different profiles of sex hormones like testosterone and stress hormones. So comparing individuals who are single versus those who are in relationships can help us understand these clusters of traits that are associated together, these psychological, behavioral, physiological traits, which presumably are associated with different life history strategies. I'm going to present first some data about testosterone. I have to give you a tiny bit of background information. Testosterone is a sex hormone that males have in much larger amounts than females. In males especially, is involved in sexual competition, so it increases when men find themselves in a situation of sexual competition, also in courtship. when a man is trying to impress a woman, trying to start a relationship, their testosterone goes up, and also in sexual interaction. So sexual arousal is typically associated with testosterone. So what we did in a study was we simply took a saliva sample from 385 business school students. We decided to divide these students into different ethnic groups. So we had a group of white, Caucasian whites. We had African-American students, Hispanic, Far East Asian, so Chinese, Japanese, and then Indian students. We found that in most cases, single men had higher testosterone than men who were in relationships, with one exception. The Far East Asian students have the reverse pattern. The men in relationships have higher testosterone than the singles. We thought, that's interesting. We did another study to see whether we could replicate this result, and we did. This time we compared men in relationships and men who were single, their testosterone levels. We had 4 subgroups of ethnicity: Caucasian, African-American, Hispanic, and Asian. The same thing in all groups except the Asian. Singles have higher testosterone. In the Asian groups, it's the opposite. So what we did then was to collect a lot of measures of sexual experience and sexual behavior in these students. For example, we asked them how many sexual partners they had in their life up to that point. And we found something interesting. Comparing just the Asians with all the others, we found that in the Asians, the singles had less sexual experience, had fewer sexual partners when compared to individuals in relationships. Whereas in all the other groups, the singles had more sexual experience. We also used measures of sexual behavior like how many one-night stands did you have in your life? The pattern is the same. So it seems that among the Asian men, those who were single, maybe it's not their choice, or maybe it's by constraint, or maybe they're single because they don't want to think about sexuality. Maybe they just want to study hard or have other things on their minds. Whereas the singles in other groups are more sexually active. Now, if we compare testosterone in all the groups except the Asians, so now we eliminate the Asians, and we look at men who are single, men who are in short-term relationships, so 3 months or less, men in long-term relationships, 3 months or more, and men who are married, you can see this nice pattern of testosterone variation, where the singles have the highest testosterone and the males who are married, the lowest testosterone. This has been replicated hundreds of times almost. It's one of the most well-established findings. Single men have higher testosterone, especially than men who are married with kids. If you're a male, you get married, you have a child, boom, your testosterone drops immediately and stays there, unless you do something about it. Same thing with number of sexual partners. Again, it seems if you compare the first 2 groups, the singles and the men in short-term relationships, overall they report a higher number of sexual partners than the men who are in long-term or married relationships. So, in summary, we believe that these data suggest that testosterone variation does not really track differences in relationship status in itself. It's not that being single or being married matters in itself, but variation in sexual behavior. So, so the men who are single might have sex more often and with more different partners than the men who are married. Maybe the men who are married have a lot of sex, but with just one person. So it's that matters for testosterone, not whether you're single or in a relationship. In general, single men have higher testosterone. But if men who are single, like the Asian men in our study, have low sexual activity and low sexual promiscuity, then they have lower testosterone. So is high testosterone a cause or a consequence of being single? It's both. It turns out that the way testosterone works is through feedback loops. So that if you have high testosterone to begin with, you're more likely to be single. And if you behave in a certain way as a single person, your testosterone goes up even further. Same thing with if you're married. If you have low testosterone to begin with, you might want to be married or be in a stable, nice relationship with just one person. That in turn will decrease your testosterone. So it's both a cause and a consequence. Now, let me tell you about the stress hormones, cortisol, and relationship status. I have to give you a little background. From the perspective of life history, differences in cortisol might reflect differences in the environment in which individuals live. So, for example, individuals that live in stressful environments are expected to have higher levels of cortisol than people who live in non-stressful environments. So, for example, if you're in a relationship and this relationship is unstable and stressful, you probably have higher cortisol levels. So cortisol filters information from the environment and usually alters metabolism. It's a metabolic hormone. Testosterone influences motivation. Our hypothesis was that single individuals have higher cortisol levels than individuals in relationships. And that's what we found in men and women in many ethnic groups, with one exception. This time it wasn't the Asians. They had the opposite partner, but the African-American men. So in the whites, Hispanic, and the Asians, the singles have higher cortisol. In the African-Americans, it's the men in relationships that have higher cortisol. And we discovered, first, this is a nice, again, picture of if you exclude the African-American and you just look at whites, Hispanics, and Asian Americans, you see a nice pattern of decline in cortisol from being single, from being married. So why are the African Americans different? Well, we asked this question. These are just men in relationship. We asked them, is your relationship stable or unstable? Well, most of the African Americans said our relationships are very unstable. So in terms of relationship instability, the African American were much higher than the other. And so we think that this is what explains why they have high cortisol level. So being in a relationship, if it's stable, acts as a buffer against stress. So marriage is beneficial if it's good quality and stable marriage. If you're in a relationship, including a marriage, and it's unstable and stressful, your stress hormones go up. We didn't just want to measure cortisol in baseline conditions. We also wanted to look at cortisol reactivity to a stressful manipulation. So in one study, we used this paradigm known as the 3-year social stress test, in which a subject, in this case a female student, is invited to the lab. She's asked to stand behind a microphone in front of 2 lab assistants who are wearing white lab coats. The test lasts 10 minutes. It's video recorded. The student is told for 5 minutes you have to talk about yourself, pretend that you're given a job interview, tell us about who you are. And for the next 5 minutes, it's a mental math test. The student is given a number, for example, 3013. She's asked, now subtract 73. From the results, subtract 73 and keep subtracting the same amount until you get to 0. If you make a mistake, we'll tell you to stop and start over. This happens to be stressful. And so if you collect a saliva sample before and one after this 10-minute test, what you see in the graph below, the red line, your cortisol goes up, especially in the 10 minutes after the test. Cortisol is a little slow in going up. So this is a good test of social, psychosocial stress. It's the stress of being evaluated by strangers. We discovered that extroverted individuals are less stressed by this. Their cortisol doesn't go up as much. And if we ask them what their social sexual preferences are, do they like monogamous relationship or more promiscuous, they report more interest and preference for promiscuous relationships. It turns out that what mediates this relationship is how much cortisol they respond with to stress. So the less they respond with cortisol to stress, the more interested they are in casual relationships. If they're more stressed, they're less interested in that. So, in summary, being single is generally associated with higher cortisol level. Maybe the single lifestyle is riskier, more uncertain, more unpredictable. High baseline cortisol is also associated with some personality differences, like being more open to experience. In general, being in a relationship is associated with lower cortisol unless the relationship is unstable. And low cortisol reactivity to psychosocial stress might explain why extroverted individuals prefer and engage in more promiscuous sexual relationships. They don't find it stressful to talk to strangers in novel situations, whereas people who are easily stressed don't like to do that. In conclusion, being single in a short-term or in a long-term relationship reflects both people's choices and the constraints that they had, but it's associated with different patterns of social sexual behavior, stress and sexual hormones. So there's something valuable we can learn comparing individuals who are single and those in relationships about the association between all these different traits, psychological behavior and physiological traits. And I think the life history perspective is helpful as a general theoretical framework. So all the work that I presented, obviously I haven't done it by myself. I had the help of some colleagues, the help of some postdocs, including Davide Ponzi, who's now at the University of Parma. He took the lead in many of the studies that we did, and a lot of PhD students as well as undergraduate students.